Hello and welcome to this presentation on bacteriophages. My name is Stephen Hagens and I would like to show you how phages can improve food safety in the food industry. I will spend a few words on Micreos, our company, but I want to focus on phages. Phages on food surfaces, which is a situation that is highly different from the situation we are familiar with, which is liquid cultures in a flask with bacteria and phage infections there, where the phages are able to completely annihilate a bacterial population. The situation on food surfaces for most pathogens is really different, and this is important to understand how phages work in theory. And after having gone through the theory, I want to talk about actual applications. Just because something works in the lab doesn't mean it works in real life, because there you have processes, complex processes that have to go very fast, but still I want to show that phages can be applied in almost every production situation. Micreos is a Dutch company. We are the global leader in applied phage technology. We do have a US sales office and we have a stock position in the US. We provide targeted antibacterial solutions. Our phage products have applications in human health against Staph aureus, in food safety, and in animal health. We have two production facilities, one in Bilthoven, where Micreos human health line of glad skin products against Staph aureus infections that aggravate skin conditions are produced, and one production facility in Wageningen where our phage products against Listeria, E. coli, and Salmonella are produced. Everything we do has a very strong background in science. We work together with a lot of universities and food institutes, mainly on application data showing that phages work under particular circumstances in different foods. But we also do fundamental research with our research partner, the Swiss Federal Institute of Technology, or ETH, in Zurich. When talking about foodborne pathogens, the biggest players have to be E. coli, Salmonella, Listeria, and Campylobacter. Depending on what type of food you produce, only one of these might be of concern to you. And we have products against E. coli, Salmonella, and Listeria, and working on our Campylobacter product as we speak. Let's have a look at the life cycle of virulent phages. The first step is absorption of the phage particle to the bacterial cell, and this is highly specific. The phage look for receptor molecules which are unique to suitable hosts. This is followed by injection of the DNA, replication of phage DNA and phage proteins, which are assembled and matured, and this is followed by host lysis and progeny phage release, after which the whole cycle can start again. Now one would think that a single phage can ruin a bacterial population in this way, and that is true, but it would happen in liquid cultures and certainly not on foodstuffs with very low bacterial contaminations. Let's have a look at the situation in Listeria. Here is a study performed by an American research group, and they collected 31,705 food samples, which they bought at retail, and they took it upon themselves not only to look for Listeria, but also to look at Listerial numbers in these foodstuffs. And what they found is this: 577 of these foods contained Listeria, but more interestingly, the majority of these foodstuffs were contaminated at extremely low levels. This is the detection threshold of 0.04 CFU per gram. And really the majority contained low numbers of Listeria, and only several of them contained levels which are considered dangerous. So this is the reality in foods. We have very low levels of contamination, a single cell in a package of ham, or maybe in 100 packages of ham. And these are the contaminations we want to destroy. This is why a critical dose of phages is necessary, and our recommendations usually are at least 10 to the 7 PFU per square centimeter on most foods. Our PhageGuard Listex product contains a phage called P100. It has an extremely broad host range within the genus Listeria. It kills all species that are commonly found in food facilities. It also has a really high affinity towards cells, so it infects easily, which is important. Plaque formation on a plate doesn't tell you everything. Infection has to be effective as well. Here you see a study on hot dogs carried out by the Swiss Federal Institute of Technology in Zurich at 6 degrees for 6 days. Hot dogs were contaminated at a level of 10 to the 3 CFU per gram. In the control, you can see that there is a lag phase of about 3 days, after which Listeria starts growing. At 2 different treatment levels, we observed a 2.5 and 3 log reduction. In case of the 3 log reduction, no regrowth occurred because all the Listeria were eliminated. At the other dose, a 2.5 log reduction was observed, and Listeria started regrowing at the same rate as the control. However, this contamination level is unrealistically high, as we saw earlier, and a 2.5 log reduction is more than overkill to reduce normal contamination background to absolutely zero. This group did the same type of experiment with a whole lot of different foods, and they saw small differences. Significant reductions were achieved on all of these products, but liquids and very smooth surfaces work slightly better than more complicated surfaces such as ham. This doesn't really surprise anyone. We all know that phages kill bacteria. So how do you apply these in real life? Let's have a look at a really simple application. This is in smeared cheeses, which are surface ripened, and it's the ripening period which is the problem. Really low contaminations can grow up to really tremendous numbers. And cross-contamination happens because these cheeses are smeared with brushes, and the same brush and the same solution is used for different cheeses. And even though there is a low incidence, you get very high numbers of Listeria in the final product. So you start with one and you start brushing them, and eventually all the cheeses in your ripening cellar will be contaminated. As I mentioned, phages work really well in liquids, so we can actually protect the smear solution against growth of Listeria. Listeria will be killed if it gets into the solution, and this protects all the cheeses. Phages work on corn, for example, and it works really fast. We saw similar reductions after 1 minute and no significant increase after 30 minutes. But industrial processes can be slightly more complicated than the smearing of cheeses. But it is no news that you can spray antimicrobials in vacuum pouches before you seal them, and the vacuuming actually makes sure that the distribution of whatever you spray in there is perfect. But sometimes low-tech options are available. You can, for example, spray phages into the hopper, and the movement of the product ensures good distribution. Whole muscle products can also be treated by sprayed lethality in container solutions. And again, the vacuuming will make sure that the phages are distributed well over the whole product. Such interventions can be highly effective. Here we have a study performed at the University of Gent in Belgium, where ham was contaminated with Listeria at about 17 CFU per gram, which is still much higher than you would ever find in real life. And this particular ham did not contain any organic acids to slow down Listeria growth. As you can see in the control, levels reach 10 to the 2 CFU per gram after about 10 days, and after 30 days we have 10 to the 5 bacteria per gram. This product would be absolutely lethal. Each treatment at 2 different levels resulted in reduction and Listerial numbers below the detection threshold for 30 days and 42 days respectively for the 2 treatment doses. Staying with RTE meats, some products are sliced, and you all know these slices. They are amazing. They will slice about 700 paper-thin slices in a minute. And treating every slice back and front is a challenge, but a challenge we accept. It is possible to spray extremely small volumes extremely fast using ultrasonic nozzles, and in this study it was attempted to treat turkey, ham and corned beef sliced with ultrasonic spraying and phages. And it worked for all 3 products. I only showed the sliced turkey data. More than 1 log reductions were achieved within 6 hours after treatment with different phage levels. Phages and organic acids, in this case buffered vinegar, can be sprayed simultaneously. This allows for much lower doses of the organic acid than you would usually require. The contaminated sliced turkey treated with phages, buffered vinegar, or the combination as a surface treatment. And what you can see that the control grew out crazily. The phages reduced the number of bacteria, but again we saw regrowth of the bacteria. And the buffered vinegar, well, it does help to reduce outgrowth, but it is not good enough by itself. The combination, however, remained under the detection limit for almost all of the 120 days, with only one data point coming slightly above. So this is an extremely interesting proposition for those who want to reduce organic acids because of organoleptic issues, or who do not want to reformulate their product. Bacteriophages work really well on food contact surfaces. The following experiment was carried out by the University of Mississippi on stainless steel with biofilms grown for either 2 days, shown in panel A, or 7 days, shown in panel B. Phage treatment resulted in a 5.5 log kill after 24 hours for the younger 2 days old biofilm, and even for the older biofilm, a 3.5 log reduction was observed. And of course, this is a one-time application of the phages. You could reapply them and get further reductions, obviously. In another study carried out at the University of Wageningen in the Netherlands, phages were shown to outperform commonly used cleaning agents on stainless steels against Listeria in the presence of food debris. These stainless steel coupons were contaminated with either milk, ham, fish, or vegetable matter before the biofilms were grown. The phage activity is shown in the last bar, and it outperforms all the cleaning agents in all cases except for the vegetable matter. These cleaning agents are highly reactive, and they react away with organic matter. So the milk and ham or fish actually eats up the cleaning agent's power, whereas phages only care for salmonella being present, yes or no. They will ignore such organic matter. Now it's time to change the subject slightly, and I'm saying slightly because we'll still be looking at phages, but we will be looking at PhageGuard S, our salmonella intervention. PhageGuard S contains 2 different phages. They work against all reservoirs of salmonella. The product is FDA GRAS approved. It has USDA approval as a processing aid and is listed in Directive 7120.1. The product is also considered organic. The 2 phages in PGS are the perfect team. They recognize different receptors which are present on all strains of Salmonella, so in theory both can infect all strains, but by using 2 different phages with different attachment sites, the bacteria cannot escape in any way. They would have to modify both receptor molecules to escape the phage intervention. And the phages also have different DNA modification strategies, allowing them to escape various defense mechanisms bacteria might have. Phage S16 is a T4-like phage. It attaches to the membrane protein, outer membrane protein C, which is a protein that is present in all strains of Salmonella, regardless of serovar. Phage F01A is a Felix O1 type phage, and its receptor is a sugar molecule that is present on the outer core of the LPS molecule, again a molecule that is present in all strains of Salmonella, regardless of serovar. Here you see the results of PGS intervention on artificially contaminated chicken skin. After contamination, the chicken skin was treated with 2 different levels of PGS, resulting in 1.5 and 1.7 log reductions, respectively. As with Listeria, the phages have no effect after the initial reduction. After about 24 hours, they get stuck. They absorb to proteins on the surface of the product. The big difference with Listeria is that at 4 degrees refrigeration temperature, Salmonella is unable to grow. So where you saw outgrowth of the control and outgrowth of survivors after treatment with Listeria, you don't see that in Salmonella. PGS doesn't only work on poultry, it also works on spinach. As you can see here, you get a 1.3 log reduction at the same treatment level after 24 hours, after which there is no continuous activity of phages. There is a slight reduction over time, but this happens in the control as well. Spinach is not a surface Salmonella appears to like very much. Some of the biggest issues with Salmonella are found in ground meat products, and this study by the University of Nevada looked at PhageGuard S reductions of Salmonella on pork trim and coarse and fine ground beef. Again, the products were artificially contaminated and treated with PGS. And you can see that on the trim and coarse grind, you get very similar reductions by PGS, whereas in the fine ground beef, reductions are slightly lower, but they are still highly significant. Up to 2 log and higher reductions were achieved in this study. The meat industry has only a few interventions at their disposal to reduce salmonella on the final product. Apart from PhageGuard S, they can use UV, they can use peracetic acid and lactic acid. PGS, the phages outperform peracetic acid and lactic acid. Even though these products are highly antimicrobial, they have a problem in meat that there is a huge surface full of proteins, and like the cleaning agents in the Listeria biofilm study, they are highly reactive and they will reactivate very quickly with all the proteins in the products being treated. PhageGuard S also slightly outperformed UV light, and the combinations of these treatments are seen below that. I won't be showing you lots of data on our E. coli phage product. The graphs all look very similar. You get reductions of 1 to 2 log after 24 hours, after which the activity of the phages stops. You get the same sort of dose response on susceptible foodstuff, and it works on all susceptible foodstuffs. Our products are considered processing aids around the world, and they are available all over the world. I would like to thank you for your attention. If you want more information on the subjects I discussed, you will find links to studies on our website and also more general information about the applications on phages in general. Goodbye.